Let's take a look at how action potential leads to contraction, or how incoming depolarization leads to the physical shortening of the contractile cell. And the first thing I'm going to make a note of here that we need to remember is that the contraction is going to be initiated by extracellular calcium. Initiated by extracellular calcium. So this is calcium that's coming from outside of that contractile cell. So let's draw the sarcolemma. So this is going to be a zoomed in version of the surface of that contractile cell. So this is if we were looking at it from the side. Here's our bilayer. Now, this is our sarcolemma, right? But this little dip here is going to be our T-tubule. So remember, that's how we access the internal part of that cell and all those myofilaments that are comprised of the sarcomeres, the contractile units. Some other things we need to draw in here would be the sarcoplasmic reticulum. And remember, the sarcoplasmic reticulum surrounds all these myofibrils and sarcomeres that allow for calcium release. So in here we have a whole bunch of calcium within the sarcoplasmic reticulum. Okay. All right, and then there are some channels that are going to help us with our concentration gradient of calcium. So the first of those I'll draw in the membrane here on the sarcolemma. Call this channel 1. And this is going to be our sodium-potassium pump. So remember, with 3 sodium inside the cell, 2 potassium. That does 2 things for us. It keeps the concentration of sodium inside the cell low, on the potassium inside the cell high. So it establishes a gradient that we can use in the next step. And this next one here we'll call 2 is the appropriately named sodium calcium exchanger, NCX. And it does just that. We're exchanging sodium for calcium. So we established this gradient by concentrating sodium outside the cell here, just with our sodium-potassium pump that we just drew. Okay. So sodium is going to want to move into this cell where the concentration is low. We'll draw that. So 3 sodium move into the cell, which can then power, so down its concentration gradient, which can then power the movement of calcium up its concentration gradient. And it's pumped outside of the cell. And in this way, we can keep the calcium concentration within the cell low. Now, there's one other way in which we move calcium. The third way. And this is just a straight up primary active transport, so ATPase pump. It uses ATP directly to move calcium against its gradient and concentrate it outside of the cell. Okay? So this is primary active transport. This one is relying on that sodium concentration, so this would be secondary active transport, okay. And this also would be primary active. Just turned 2 the other day. Okay, so just to review here, sodium-potassium pump concentrates sodium outside of the cell. That sodium wants to move down its gradient, and then moves through this sodium-calcium exchanger down its gradient, which allows us to power the movement of calcium against its gradient and concentrate it outside the cell. The other thing we can do, we can use this calcium ATPase pump, use ATP directly to move calcium against its gradient and concentrate it outside the cell. So basically here what we're creating is this large extracellular gradient of calcium outside the cell. Remember, this is important because this contraction is going to be initiated by extracellular calcium. Okay, let's draw some other things here. So let's just say we have an incoming depolarization. So we have an incoming action potential. This can be from other contractile cells or maybe an autorhythmic cell. Doesn't matter. We have incoming action potential. Now, that within these T tubules, that is going to reach a specific protein here. And the one we're interested in is going to be those L-type calcium channels. So just go back and look at the membrane potential drawing we just did. It is the L-type calcium channel that kind of starts this, allows for calcium to slowly enter the cell, contractile cell, for a long time, which gives us that plateau phase. So action potential reaches this L-type calcium channel, and we're going to see an influx of calcium. So this is that extracellular source of calcium. That's a voltage-gated channel. It's going to be triggered to open up. Calcium is going to move in. I'll add voltage-gated to that just so we're clear. Now, where does calcium go upon entering the cell? Well, upon that sarcoplasmic reticulum, remember, we have a certain receptor, and that is called the ranitidine receptor. RYR. Ranitidine receptor. So calcium goes and binds the ranitidine receptor, and that allows for release of calcium from the sarcoplasmic reticulum. So this is called, that calcium leading to the release of more calcium is called calcium-induced calcium release. So we have incoming calcium that induces the release of more calcium from the sarcoplasmic reticulum. And it works out that about 20% of the calcium source is extracellular, so coming in here, but it leads to more calcium release from the sarcoplasmic reticulum. 80% of the calcium that is ultimately released that helps with the contraction. Okay? So that is calcium-induced calcium release, a term you should be familiar with. But it is extracellular calcium that kicks off the whole thing. Okay, so now we've released calcium. And what is that going to do? Well, remember, this is releasing it right next to those myofibrils, those contractile units, the sarcomeres. Remember, that's comprised of thick and thin filaments. Thick filaments here, sarcomeres. And it is this calcium here that leads to shortening of these contractile units. Remember, we need calcium to bind, to move the tropomyosin out of the way to allow for contraction. Okay? So these are our myofilaments here. And when those sarcomeres shorten, the entire contractile cell shortens. Now, at the end of the contraction, we need to get calcium back into this sarcoplasmic reticulum. So here we have the Circa pump. you're not familiar, that stands for sarcoplasmic endoplasmic reticulum, calcium, ATPase. So with that ATPase, we know we're going to directly use ATP to move calcium back into the sarcoplasmic reticulum using ATP. Okay, so just to work through the entire process one more time from start to finish, we have incoming action potential, incoming depolarization. That depolarization activates our voltage-gated L-type calcium chain. That opens up, and we've been concentrating calcium outside of the cell via these mechanisms here, right? That allows for a calcium concentration gradient. Calcium wants to move into the cell. When it does, it binds to the ranitidine receptor. Binding of the ranitidine receptor causes more calcium release from the sarcoplasmic reticulum. Okay? This is calcium-induced, calcium released. 20% incoming calcium from extracellular sources leads to 80% release from sarcoplasmic reticulum. Calcium comes along, binds the troponin, pulls the tropomyosin out of the way, and allows for interaction of contractile units or contractile components, and we get physical shortening of the cell, or contraction. Okay? At the end, we get that calcium back into the sarcoplasmic reticulum via the Circa pump.